Namaste, I am Dr Garima Gupta, and today we will be discussing that what is the importance of blood stains as an evidence in forensic science. So in this we will be dealing with the history of blood analysis or blood pattern analysis. Then we will be dealing that how the analysis of blood stain is done, either by visualizing them or by doing certain kind of special test which proves that it is a blood or what kind of blood. So moving to the next slide. Now the history of blood pattern analysis. Blood pattern analysis is mainly based on two basic principles of forensic science. One is Locard principle of exchange, which was given by Professor Dr. Edmund Locard. He was the pioneer in forensic science. Actually, he told that whenever a person comes into the contact of a site or a place, it leaves certain traces in that particular place, and he might take some things from that particular place and can keep with himself, and these things which he has left or taken can be claimed as clue in the forensic science or finding out the criminal. So it simply means the contact leaves certain traces which can act as clue for determining the criminal. Now the next principle is principle of individualization, which simply means that every individual has certain characteristics which is special for that particular person. Like for two persons the DNA could not be same. Every person has a unique DNA, or there are certain proteins or enzymes which are particular for a person or a individual. So on the basis of these individual things which are special for a particular person, we can identify that particular person involved in the crime. So the whole blood pattern analysis is based on these two basic principles of forensic science. Now the first paper on blood spattering was published in 1895 by Dr. Edward Piotrowski. Then another scientist, Dr. Victor Balthazar, worked on the same topic. Afterwards, from 1971 to 1982, Professor Herbert Leon McDonnell published three papers, and several books on blood spattering. After which, in 1901, Karl Landsteiner found out about blood typing, and it has been a very successful and good method to identify the blood groups. Now, what is blood? It is a slightly alkaline fluid which circulates around the body, and it helps in feeding the cells or transporting the oxygen and waste. It contains two parts. One is fluid part and the other one is solid part. So fluid part contains mainly the plasma and the serum, while the solid part or the non-fluid portion consists of red blood cells and white blood cells. Now it is composed of mainly five things: water, cells, enzymes, proteins and other inorganic substances. Blood undergoes various kind of changes with the passage of time, like the cell structure might get destroyed, or there might be loss of activity of protein. There is color change, and as well as physical and biological factors of blood get affected. So blood stains are very important in forensic science. They might be involved in various kind of criminal cases like physical violence, murder, assault, etc. Blood stains may be present on weapon, murderer, crime site, or even after washing they can be found in trapped fibers of cloth. Although blood color may change due to the reaction with chemicals and paints, so blood stain should be checked properly by various methods, which we will deal later. Now, what are the precautions which has to be taken while collecting the blood stain from the crime site? The examiner, forensic examiner, should wear protective clothing like gloves, masks or eye protection. Then the photographs should be taken from all the angles. Photographs are very important as they may help for solving the case in the later phase. You can see the pattern of blood at that particular time, and you can claim that this particular blood stain was present at that particular site. So photographs are very important. Then the victim body or cloth of victim should be carefully preserved without any contamination. The contamination of any kind should be avoided. If the victim's cloth is cut for taking the blood stain, then it should not affect the area of stain or the bullet wound, as it may interfere with the ballistic analysis. The stain should be dried either in fan or in shadow. There should not be any kind of moisture in the blood stain, otherwise molds may grow on it. Then the packing should be done in polythene, and there should not be again any kind of moisture, as it may lead to bacterial growth and damage the stains. For one stain there should be one cloth for wrapping. The two stains should not come into a contact, because the two stains might be of different origin, and if they will come in the contact, they might contaminate the analysis and the stains. So the analysis won't be done properly. So for each blood stain, a separate wrap or a separate clean paper should be inserted for preserving the blood stain. If there is any kind of liquid present on the crime site, then a dropper should be used, and the liquid blood should be kept in a vial under ice cold condition. Or a piece of sterile cotton or filter paper dipped in saline water can also be used, and this can be further dried. If the blood stain is present on a big unportable object, then that particular area containing the blood stain can be cut, or a blood stain can be scraped from that place, and then it can be kept on clean paper and further preserved. For any object containing the blood stain, a control should always be taken so that it won't interfere in the blood pattern analysis or various other tests. So a part of object which is not containing any kind of blood stain should also be taken as a control. For collecting the blood stains, it can be easily collected in 10% solution of potassium cyanide or in 10% solution of glycerine made up in distilled water, or a weak solution of ammonia can also be used. For collection of sample, the stain area can be easily rubbed with the cotton swab, which should be moistened with distilled water. Then the swab is air dried, not heated. Then it is simply placed in a sterile swab tube, labeled and properly sealed. All the swabs which are collected, that should be arranged in a sequence as they were collected, and that should be kept properly labeled. Now, to preserve the blood, it can be preserved in various solutions, like it could be preserved in sodium fluoride solution, potassium oxalate, lithium or ammonium oxalate, EDTA, citrate, sodium hydroacetate, and all the preserved blood samples should be kept at 4 degrees Celsius. Now, what are the informations which can be drawn from observing the blood stains at a crime site? The first thing which can be detected is the direction of droplet which was travelling from the body to the surface or wherever it is present. Then the angle of impact of blood droplet on the surface. Then it can also be used to identify the probable distance of droplet from the target from which the blood droplet has come, means the body. Then what was the nature of force involved in making the blood stain to come out, and what was the nature of object used to do the crime or to take out the blood stain. Then what was the relative position of suspect, victim and the other related objects. It can also be used to identify the number of blows which was done to do a crime. It could be a death or anything which has happened on that particular crime site. Then it can be also used to find out the sequence of events which has. Now from these informations, mainly two conclusions can be drawn out. First is, we can easily find out the events and sequences of the crime. Assumptions can be made on the basis of position of victim. It was standing, sitting or lying, or what was the struggle. By observing the blood smears and blood trace, it can be assumed. The second conclusion which can be drawn is that the suspect, victim or the witness, whatever they are making statements, they are matching with the blood pattern or not. So it can be matched that the statements are matching with the blood pattern obtained at the crime site or not. So it is very informative. Thank you so much for listening.